Gabrielle, you are the first scholar supported by a gift from the Secunda Family Foundation. Would you tell us exactly what you're studying?
I am studying the impact of glucocorticoids on the central regulation of appetite and energy balance.
Steroids, you mean?
Steroids, that's right. About 1.2% of the American population is actually treated with steroids in a chronic way for an array of conditions. The downside of the medications is that they have adverse systemic metabolic effects. And one of the things that happens when you take steroids consistently is that you tend to gain weight, and they really contribute to the prevalence of obesity in this country. We research the neuroendocrine regulation of appetite and energy balance and how those regulatory processes are impacted by differing hormonal states.
So has this been a useful grant for you in terms of your research?
Useful is really an understatement. I mean, this has been a tremendous award, and the funding that we've received has allowed us to support the use of state-of-the-art energy calorimetry chambers. It's allowed us to purchase really sensitive neuropeptide assays, and it's provided support for the technicians and for the equipment and machinery that we use to sort of measure peptides in blood. And having funding from the Secunda family allows us to sort of be more adventurous in what we're doing, to think out of the box, to innovate. So, I mean, at this point in time, what we're interested in figuring out is the differential effects of glucocorticoids in men and women. One thing that we know from animals is that knocking out the glucocorticoid receptor on AGRP neurons has differential effects on male animals and female animals.
Is that so?
Yes. Female animals are much more responsive to having the glucocorticoid receptor knocked out. So when you take that out of the equation, the animals increase their energy expenditure manifold and they lose a lot of weight. That happens less in male animals. And I think that has to do with sort of the evolutionary response to cortisol. In settings of stress, when high cortisol levels are around, women tend to actually hang on to adipose tissue. In the setting of stress, in comparison, in human men, they actually expend energy and burn calories in response to stress. And it's quite surprising that there's really very, very little data out there now on the differential effects of glucocorticoids in men and women.
Well, we sponsor gender-specific investigation because we think it's very important.
Absolutely. And I think one of the things I definitely value about the foundation and the emphasis on gender-specific medicine is I think it's really the critical first step towards personalized medicine.
A good observation by you. That's exactly right.
Absolutely.
Many, many people have said to me, when you have personalized medicine, why do you need to know whether a patient is male or female? And the answer is because genes are differently regulated in males and females, and that's not examined in experimental work. So I'm glad we hit home with you.
Absolutely.
Jennifer, tell us about what this grant has meant to you.
So this grant has been very exciting for me, especially at this time. I'm starting my own independent laboratory right now, and so this funding has been crucial to start up our projects and to obtain some of the supplies that are required to start a new laboratory. And I have a very interesting mix of collaborators. I'm working with Dr. Landry, who is head of medicinal chemistry and chair of Department of Medicine, as well as Ottavio Arancio, who is a neuroscientist, actually studies Alzheimer's disease. And together we're developing a library of novel drugs that would potentially be beneficial in both Alzheimer's disease and also certain subtypes of lymphoma.
Tell us about your interest in lymphoma.
So I became interested in lymphoma in medical school. I was really fascinated by the biology. Lymphoma is a disease of the white blood cells, and these cells originate from the bone marrow and they travel to your lymph nodes, sort of naive. They're presented little bits of bacteria and viruses from the environment. And part of their job is to make perfect matches to those bacteria and viruses to fight them. And these are cells that ordinarily are supposed to die if they don't make a match. But sometimes there are changes in their genetic controls that tell them to keep living even though they haven't made a match. So I became really interested in how our own natural biology can sort of go awry and lead to a cancerous state. These diseases arise not from anything that we've been exposed to or what we've done, but really by following the natural biology of the lymphocyte and sort of going on hyperdrive, if you will.
I think you told me that you can look at the particular individual derangements in gene control and decide how to compose a therapeutic palette for these patients. Is that correct?
That's right. In a certain compartment of the lymph node called the germinal center is where this reaction occurs. And we see that there are lots of mutations that coalesce to lead to this gene regulation. So we're looking at specific changes in individual patients to see how we can target those mutations to allow for more normal behavior of these cells to prevent lymphoma and to treat lymphomas. We're finding that specific mutations occur in specific diseases, and we can identify those and then use targeted therapeutics to direct treatment directly at those mutations rather than treating patients with chemotherapy, which can be very toxic. As a translational researcher, I'm studying biology in the laboratory, but we're also running clinical trials to take some of these findings directly to patient care, to try to enhance their quality of life, and that's been extraordinarily rewarding for me. So we're not using chemotherapy so much, but rather targeted therapies, which will preserve fertility and their overall well-being.
Chen, you are a second-time awardee of our Birch-Derchin Scholarship, and I think they would appreciate knowing, as would all of us, what the investigative support has meant to you.
The Birch-Derchin Scholarship has helped me tremendously in the last 2.5 years. Inside the lab, it directly helped me to buy the reagents and equipment that I needed for my research. And this research is to figure out how the brain differs between males and females on a biological level. As a molecular biologist and a cellular developmental biologist, I do look at a lot of genes that exist in all of the animals,
yes.
And these are the conserved genes. They exist in pretty much all animal species, me included.
So now you work on the lowly worm.
Yes.
Tell us why you chose the worm as your subject.
The worm is a very interesting organism. The worm has a really complicated nervous system that we can use a lot of tools, including molecular tools, genetic tools, and cellular tools, to sort of understand on a biological level where certain genes are expressed on a single cell level. If we try to see how these genes are regulated on a genetic level, they're quite different. What I found is that these genes exist in both sexes, but during sexual maturation, only the male animals start to express these genes. And that means a whole lot because we know that the female worm and male worm actually do have a lot of different behaviors. And the behavior I am interested in is actually locomotion. You use a video recorder to record how the worms move on a little petri dish for a long time, and you quantify that behavior. You can actually find there are a lot of movement differences between the males and the females. I essentially was able to find these group of genes are expressed only in one sex, which is the male, at a certain time point during their development. If you deplete this gene from the genome that get expressed during sexual maturation only in the males suddenly just become not as active anymore. So this means this gene is really important for contributing to at least part of the sexually different behaviors. In the first 2.5 years using the scholarship, I was able to answer the first 2 sub-questions of my grant proposal. Where are these genes expressed? And the second question is, when are they expressed? Now it comes to really exciting time to figure out what are these genes doing to contribute to the sexually different behaviors, and how are they doing what they're doing? And even more importantly, how are these genes regulated? So the next step will be to deplete some of these genes that showed sex-specific expression earlier to see what happens. That's the basis of why we can use a little worm to understand the biology or biological mechanisms behind all these sexually different behaviors to give us insight into what higher organisms, including ourselves, behave.